Well, welcome to another Solanaceae seminar online this Friday. And today I'm very glad to introduce Dr. Lynn. She is a PhD. She finished her PhD at the University of California, Berkeley in 1995. And her dissertation project was in Licianthes, Ceres Masonodonte, a group of perennial herbs centered mostly in Mexico. And she has been working 25 years as curator of the herbarium at UC Davis. And she retired recently in March 2020. And now she's working on rare Californian plants for the California Native Plant Society. She has published an article in Phytokiss very recently in December 2020 on the 53 Licianthes taxa of Mexico and Guatemala. It's a great article that you should all check. So with that, I'll give you the word to Ellen to talk about the diversity of Licianthes in Mexico and Central America.
So does everything look okay?
Perfect, yes.
Okay, so thank you for inviting me to give this talk. It's 8 o'clock in the morning in California, and I don't live in Davis anymore. I moved to the beach. How about that? So I live in Ventura, California now, but I can still be reached for the next few months at my UC Davis email, which I'm giving on this first slide. And I think at that point, it will be forwarded to my personal email. And if you have any questions at all, unfortunately, I do have a work meeting right after this at 9 a.m. But Lynn Bohs, who's the co-person on this talk and provided a lot of the slides and helped me edit this presentation, we decided I would just give it. It's also very similar to a talk I gave last year at the UC Berkeley Botany Lunch. And a few of you were there, and so I'm just warning you, it's pretty much the same talk. And so if you have something better to do, you are welcome to hop off. Just to orient you, Lysianths has between the second or the third largest number of species in the Solanaceae. It's kind of tied with Cestrum. And in terms of its distribution, it's present in both the New and the Old World. As you can see from this map, in the New World, it ranges from Mexico down to Argentina. And in the Old World, it's mainly in Eastern, Southeastern Asia, and into Malaysia, Indonesia, New Guinea, and there's one species in Northern Australia. So in terms of the taxonomic history, until the very early part of the 20th century, Licianthes was included in Solanum. And it was first and last monographed in its entirety in 1919 during the First World War by the German botanist George Bitter at the Berlin Herbarium. And he divided the genus into 189 terminal taxa that were placed into 4 subgenera, 7 sections, and 15 series. As was normal back then, there are no identification keys, and the work is in German and Latin. It's been included in many regional treatments of floras, for example, flora of China, Veracruz, Mexico, Guatemala, Costa Rica, Panama, Venezuela, Peru, and Argentina. And in his introduction, Bitter discussed at length the similarities that he saw between Licianthes and capsicum, concentrating mainly on the case structure. And so he thought they were very closely related. And in phylogenetic analyses from like the last 20 years or so, the trees have always shown Licianthes and capsicum to be sister taxa, as shown in this one from 2013. So let me just talk a little bit about the calyx in these genera. So in many Solanaceae, such as this one right here, Solanum, you have a calyx that has these sepal lobes coming off. And in Capsicum and Licianthes, there are no sepal lobes. The sepals have been truncated into this rim, which sometimes is kind of membranaceous at the top of this cup. And below the rim, you can have these appendages, which are sometimes called teeth, coming out. And they're not always present, but when they are present, you often have them in the number 5. So 5 appendages, that's very common in Capsicum, less common in Licianthes, or 10, that's more common in Licianthes. But you can also have as many as 20 or 25 in some of the species of Ceres Prolifera in South America. So what are the differences between Licianthes and Capsicum? So here's a very nice slide Lynn put together. It shows Capsicum here on the right, and you can see the anthers, and you can see that the anthers are dehiscing by longitudinal slits. Whereas over here in Licianthes, you might be able to see these teeny tiny terminal pores. So all of Licianthes dehisce with porocidal anther dehiscence. In Capsicum, the stamen length is equal among the stamens. In some Licianthes, you can have unequal stamens, as shown here, where you have 1 long one and 4 short ones. At least some species of Capsicum have capsaicinoids in their fruits, and in Licianthes, you do not have this chemical. Capsicum is just found in the New World in terms of native species. I mean, of course, it's been taken everywhere. Licianthes is found in terms of native species, both in the New and the Old Worlds. So Lynn Bohs put together a team to investigate this relationship between Licianthes and Capsicum, and he received funding in 2016 to do this project, which is both monographic and phylogenetic in scope. And the team that she assembled included myself to do the Licianthes of Mexico and Central America, Gloria Barboza to do the genus Capsicum in its entirety, Sandra Knapp at the British Museum to work with us using the Solanaceae source website. And she's also working on the Old World Licianthes. Daniel Spalling, who was Lynn's postdoc for the project. And then I wanted to give a shout out to Shirley Tucker, who provided extra funding to the University of California, Davis, which helped us with hiring assistant curators so that I was freed up to work on this project. And here's the Solanaceae source website that I just mentioned that Sandy Knapp curates. And we have put our Licianthes information up on this website. So the species descriptions that I completed are here. And here's a phylogenetic analysis that Lynn completed that included 17 species of Capsicum and 36 species of Licianthes, and she can answer more questions about this analysis. And she will be available for questions at the end of this talk as well. And this is based on nuclear ITS and waxy genes and chloroplast NDHF and TRNTF genes. So I'm going to be using this phylogeny as a backbone for this talk. And I just wanted to point out, which is pretty obvious here, that Licianthes is coming out in 2 clades, what she calls clade A and clade B. And clade A, you may notice, is actually joined to Capsicum with weak bootstrap support of 59%. And then clade B is monophyletic. And so this may cause taxonomic nomenclatural problems in the future. We'll have to, she wants to get more data to try to get more information on the backbone of this phylogeny. So in terms of the grant results for UC Davis for our Licianthes work, I made 3 trips to Mexico in 2016, 2017, and 2019, but I'd also made other trips to Mexico going back to 1990. One trip to Guatemala in 2017, I was able to visit herbaria both nationally and internationally. And I completed 70 taxonomic treatments for the taxa from Mexico to Panama, and those are now posted at Solanaceae source. I also, with my team, published 10 papers on Licianthe since 2016. And this included what I call my 330-page, 30-year Ellen Dean brain dump, which was published in Phytokeys in December 2020 on the Licianthes of Mexico and Guatemala, because those are the species I know the best and I've seen in the field, and I felt like I wanted to include in this article. But we did do research on species going down all the way to Panama. So here's the link if you want to look it up. It is open source, and it includes references and information from all of the papers that I've published, and it has a very complete bibliography. And if you can't get a hold of any of the publications that I cite, just email me and I will try to send them to you. So here's the team that worked with me at UC Davis. It included 3 assistant curators that Shirley Tucker supported with funding, and Genevieve Walden, which was supported by the grant for a short time as a postdoc, and she really got me going with gathering my data and organizing it. There were 2 undergraduates, Hannah Kang and Myra Huerta, who are on the papers. There were other undergrads as well who helped with data entry and are on some of the papers. Myra in particular helped with Ceres Prolifera. I'll talk about that work in a little bit. And Hannah helped with those Panamanian species that were newly described, as well as other work. Marco Antonio Anguiano Constante at the University of Guadalajara in Mexico has been working on Licianthes for, I don't know now, maybe 5 years. Started as an undergraduate and continued with his master's and is now doing his PhD. And he concentrated first on the biogeography and molecular analysis of Licianthis mussiniana, which I'll talk about in a little bit, and now is looking at Ceres mesonodontae as a whole. And he had already started entering a lot of specimen data into an Access database when we were getting going, and he very generously shared his data with us. And we imported it all into another Access database that my husband, Tom Starbuck, put together. And by the time we finished, we had about 10,000 specimens in the database. And we gave those to Sandy Knapp, and she imported them into her big Solanaceae database and has it all organized. And there were the institutions that were involved in this work included, Missouri Botanical Garden, where Bill Darcy mentored me in the early stages of my graduate and professional career. Michael Nee at New York, who was just a huge help and very generous with sharing his data with me. Maxu helped me many times over the years with specimens, and in 2017 with field work in a field vehicle, Rafael Torres and Oscar Hinojosa especially were helpful. Victoria Sosa at Jalapa has helped me for 30 years with access to Mexico and people. And Mario Valise in Guatemala was incredibly important to my work. And here we are at the University of Guadalajara. Here's my husband, Tom, and Marco together looking at the database. So there are many other people who helped, but these are really important people. So in terms of the talk, I'm going to first go over the morphology of some of the characters that I looked at. And then we're going to do a really fast drive-by glance at the morphological diversity in this beautiful genus. And I apologize for the speed that with which I'm talking. So most of the species I looked at in Mexico and Central America are shrubs and vines. There are a few epiphytes that are present, especially in section Synanthoroides that we'll be going over. There's a minority of species that are herbs. Some of them are in Ceres Mesonodonte, shown here, which I'll be going over. And there's also Licianthis acerifolia, which is a South American herb. And what's interesting is in both of these cases, they have edible fruits. Most Licianthes have paired geminate leaves, as shown here, with a large leaf and usually a smaller minor leaf. In some cases, the minor leaves are missing, which can make it kind of confusing. You're trying to figure out what's going on. I found pubescence to be super important, and there's different kinds of trichomes. So they range from simple, so they can be straight or bent. They can collapse upon drying, with sometimes the cells oriented at 90-degree angles when they collapse, or sometimes they remain very cylindrical upon drying. They can be glandular, some of these simple hairs. The branching ranges from simple furcate to dendritic. And then there's different kinds of stellate hairs, and true stellate hairs are only found in one species, Licianthis hypoluca, where all of the rays are coming out at a single node in one plane. Usually they're multangulate stellate hairs with the rays coming out at different angles. And then you can have this other type of stellate arrangement where you have rays coming out at subsequent nodes, and I called this geminate. And then you can have this very complicated type of multangulate hair where the multangulate branches go on to be further branched, either in a multangulate way or in furcate way, and we just call those crazy hairs. That's they get pretty complicated. In terms of the calyx, you can have this type, which I showed you before, where you just have the rim and either very poorly developed appendages or none at all. You can have little tiny bumps coming out of the calyx. And just to point out, this species, Licianthis Peduncularis, is one that has the glandular pubescence, and you can see it coming out here on the calyx. This is very, oh, sorry. I'm trying to use my mouse and my, it's very sensitive. This is very typical of the appendages, most Licianthes, where they're kind of short, coming out and very pointed. You can have very, very elongate appendages, as shown here in Mariovillaceae. In a minority of the cases, you can have a very well-developed calyx sleeve beneath the rim. Usually it's, in more typical Licianthes, the sleeve is just about 0.5 millimeter long and you can barely see it beneath the appendages. But in this case, you can see that the appendages are kind of turned downward and revealing this very long sleeve, which can be 1 millimeter to 2 millimeters long. And then another thing that's rather rare is these appendages that are kind of obovate. Rather than pointed, they're wider at the tip than at the base. And that's typical of this group that includes Licianthis Heterocleta. In terms of the corolla, it's important to mention that many of the species of Licianthes in Mexico and Central America have diurnal movements. So they open and close each day. When I collected Mario Vallecii, Licianthis Mario Vallecii in Guatemala, this is how the corolla looked. It was closed in the field in the afternoon. And so then I put the material in a plastic bag with some water, and in the next morning, the corolla was open in the bag, and then I could press it and photograph it. So this technique was described by Michael Nee in the Solanaceae newsletter in 1981. And I've been using it for 30 years to get good Licianthes specimens. Most of the specimens that you see in herbaria of Licianthes have closed corollas. And so it's a pain to dissect them open to look at the anthers and the gynecium and such. And it makes it hard to give corolla diameter as a measurement in the taxonomic descriptions. And so I usually just use corolla length. So this is the diversity in corollas when they're open. So you have some corollas that are entire in outline and open all of the way, which I call rotate, it's plate-like. You can also have corollas that are entire but campanulate in outline or reflexed. You can have differing amounts of what I call the interpetallar tissue in the corolla connecting these corolla lobes. And so you can start having the lobes protruding beyond that tissue and getting the corolla becomes stellate in outline. And so you have less and less corolla interpetallar tissue until you don't have any, and you have these very deeply stellate corollas. And something that can happen in Licianthes because of these diurnal movements is that the size of the corolla can change dramatically from the first day that the flower opens to the last day that the flower opens. So it opens successive days over 3 to 5 days. And each time it opens, the cells on the inside of the corolla elongate. And as it closes, the cells on the outside of the corolla elongate. And this causes the corolla to grow in size, similar to what happens in tulip or crocus. The other thing I wanted to point out on this slide is stamen arrangement. So you do have species with equal stamens, and they often have very, very short filaments, and they're mostly anther. And then you have species that have unequal stamens with often 4 short stamens and 1 long stamen. And this is due to a difference in the filament length. And similar to the way the corolla grows over time, the stamens can change in length over time. And so you can see here that the longest stamen is much shorter on this first day flower than in this 3rd day flower right here. In terms of the pollinators of Licianthes, there haven't been that many published observations. It has been observed in Belize and Costa Rica, and also I did some observations in Mexico, and it was found that solitary bees and halictid bees are pollinators. And the anthers do need to be buzzed by a bee to get the pollen out of the poricidally dehiscing anthers. In greenhouse trials, all of the species studied, appeared to be self-compatible, but the pollen tube studies that I did didn't conclusively show where the tubes were blocked. They went all the way down the style and they entered the ovules, and then the ovules aborted. In terms of the fruits, a very common fruit coloration is what is shown here in Licianthis Heterocleta, where the immature fruits are green, they turn to yellow and orange and then red. There are some species where the color remains green, such as Licianthis mosiniana. And then there are a minority of species that can have this blue-black coloration. In terms of the interior of the fruit, most of them have kind of a mealy interior where the seeds are. In Taxonomic, which is only known from one collection from Oaxaca, it appears to have a completely dry fruit. In Licianthis Prolifera, you can see that there's kind of a spongy texture. In this one, Licianthis ciliolata and relatives, there's a powdery placental area and then a juicy mesocarp, whereas Mosiniana is juicy throughout. And in a minority of Licianthe species, you have these stone cells, which are in the mesocarp and are aggregated together into what we call sclerotic granules. And so they are coating the outside of the mesocarp. In some species like Licianthis Passiflora, it's reduced down to just one or two stone cells at the very tip of the fruit. In terms of the seeds, most of the seeds in Licianthes are kind of nondescript, flattened, with little surface sculpturing, ranging from sort of triangular to ovoid or reniform in shape. These are seeds in Licianthes Ceres tricolor. But in a minority of species, they can become quite three-dimensional and dark in color, with surface sculpturing and even fibrils sticking out of the walls. So these are Ceres Mesenodonte species. These are species in Ceres Prolifera, and this is Licianthis textiliana, which has quite a bit of surface sculpturing, but no fibrils. So I'm going to now take you on a whirlwind tour of the species that I looked at. I'm going to start with my favorite group, and Marco Anguiano's favorite group as well, which is the species of Ceres Mesenodonte that I studied as a PhD student. So on this phylogeny, they're represented by Acopulsensis, Ciliolata, Jandowskii, Mosiniana, Peduncularis, and Dejecta, and the others are Hintoni and Starbuckii. There's 8 altogether, and they come out here in Licianthes clade A. So they're perennial herbs. Most of them are endemic to Mexico. A couple go down to Central America. They have rotate corollas, single-flowered inflorescences, simple trichomes, enlarged tuberous roots, which are very unusual in the genus, unequal filaments, large green to purple fruits, large three-dimensional seeds, and one species with sclerotic granules. And here's just a little photo montage. So you can see the entire corollas. This is Acopulsensis. Some of the species have these dark purple fruits, others remain green. One species, Lesianthis Peduncularis, is prostrate on the ground and has these fruits with the sclerotic granules. So a number of these species are used for edible fruits. They're not domesticated or planted from seed, but gathered where they volunteer in cultivated fields of like corn. And the best known among them is Licianthis mosiniana, which I divided into 3 varieties, Mosiniana, Margaretiana, and Oaxacana. And as I said, Marco Anguiano has continued work on this species and has published his master's thesis earlier this year in the Biological Journal of the Linnean Society. It's a great paper. He looked with molecules and confirmed the 3 varieties, which was nice. And Lynn Bohs wanted me to mention that Margaretiana was named after my daughter Margaret, who delayed the publication of my PhD thesis for 10 years because it was just hard having a baby, you know. Okay, so the next group I wanted to go over is this group of South American species, which is represented in our phylogeny by Fasciculata, Saltensis, Rantinettii, and Lycioides. And it comes out sister to the group of herbs that I just talked about, Ceres Mesonodonte. So we're right here in clade A. And though this is South American, I did cover Licianthis rantinettii in my monograph of Mexican and Guatemalan species because it's a very well-known cultivated plant. And so I included it in the monograph. So this is Licianthis rantinettii, which is cultivated worldwide. In the United States, we call it blue potato bush. I don't know why. And it has these gorgeous purple flowers, which remain open all day and close for the night. When the fruits are immature, they're green with black stripes. And boy, they look so much like the fruit of Lisianthis Peduncularis, which is that prostrate herb that's endemic to Mexico with the sclerotic granules. When they mature, they're orange in color. And it's part of this species of South American, it's part of a group of South American species that Bitter called Ceres Prolifera. And all of these species have the stamen arrangement with 3 long and 2 short stamens. And then they have the sclerotic granules in the fruit. This is Licianthis fasciculata, and this is Licianthis lycioides. These photos are from Michael Nee. And Licianthis lycioides is type species of the genus. And it was placed in a different subgenus, Licianthes, by Bitter because the sclerotic granules in this case become actual pyrenes that are surrounding the seeds. So it's a little bit unusual. Finishing our tour of clade A, I just wanted to talk about Ceres Strigulosae, which was a group of species that Bitter singled out and is now being worked on by Marco Cueva Manchego. And he gave a talk in this series a few weeks ago, an excellent talk on his work on this series. And it's available. I'm giving you the link right here on YouTube. And he showed that the species fall into 2 clades, and there are more species and are recognized currently, and he's working on that. So this group of interesting species that's mostly South American are shrubs and subshrubs with strigose pubescence with simple trichomes, geminate, usually very unequal leaves, stellate corollas, some of which may lack diurnal movements, equal filaments, and no sclerotic granules. So for my work on the species of Mexico and Central America, I mainly was dealing with these 3 species. And I had to deal with this mess that is Licianthis amatitlanensis and Licianthis inaquilatera. So Licianthis amatitlanensis is a species that was described from Guatemala as present in southern Mexico and goes down to South America. And Licianthis inaquilatera is a species that was described from Bolivia and is found in South and Central America. And those 2 species overlap in Costa Rica and Panama. And some people have called them the same thing. So there's a lot of specimens that are just identified, everything is identified as inaquilatera. And Lynn Bohs suggested I look at the trichomes more carefully. She felt like there was more than one thing there. And so we did an analysis of the morphology of the specimens and the trichomes. My undergrad, Myra Huerta, helped me with this. And we felt like we were seeing a difference in the orientation of the strigocombs. So this is a picture by Marco Cueva of Amatitlonensis, and you can see the orientation of the trichomes sticking out from the stem, very erect. So it's at a 90-degree angle from the midvein of the leaf, and it's very erect on the stems. And an herbarium specimen, this is how it looks, where you can see the trichomes sticking out at a 90-degree angle from the veins of the leaves. And this is in Aquilatera, where the trichomes are very oppressed and softer. We also looked at elevational differences and felt that Amatitlonensis is present in lower elevations, at least in Mexico and Central America, than in Aquilatera. And where these two overlap in Costa Rica and Panama, we did see some intermediate specimens at intermediate elevations. All of these have the same one-flowered inflorescences with very short pedicels that hang underneath the leaves. And then there was this third entity in Central America, which was described from Guatemala as Licianthis inconspicua, and very high elevation cloud forest. And it has much longer pedicels on the flowers and the fruits than Amatitlonensis and Aquilatera. And we grouped the material from high elevation cloud forest with these characteristics into one species, Licianthis inconspicua. It may be that the material from Costa Rica and Panama should be called Licianthis storkii or a different name. So I may have gotten that wrong, but at any rate, they're very similar. And then we also described a new species from Mexico in this group, Licianthis glabripetala. And it's in cloud forests in central Mexico into northern Veracruz, and it's quite separated geographically from low elevation Licianthis amatitlonensis, which is in southern Veracruz and down into southern Mexico. And the hairs on the back of the leaves look quite different. So this is Amatitlonensis, and the glabripetala trichomes are quite curly and almost tomentose, and the corollas are larger and they're glabrous. So based on habitat, elevation, and morphology, we separated out this new Mexican endemic. Another group we spent quite a bit of time on is Licianthis Prolifera and Licianthis Ceres Prolifera, which is a group of species that Bitter put together. And it's named for Licianthis Prolifera in Oaxaca, Mexico. And it's one of the black purple fruited species. All have simple trichomes, colorful corollas with interesting markings, and equal unfused stamens and sculptured seed surfaces with fibrils. So it's got quite a few characters uniting the species. This down here is Licianthis quicensis. This is Licianthis prolifera. This is the seed surfaces of Licianthis prolifera. Quicensis has a red fruit. Prolifera has a dark blue fruit. Prolifera is from Oaxaca, Mexico. Quicensis is from Guatemala. And this is Aaron Rodriguez, who gave a talk, was it last Friday or the Friday before that, on the diversity of Solanaceae in Mexico. And you can see from his outfit that it was quite cold where we were collecting Licianthis prolifera in the high elevation Sierra de Juarez in Oaxaca. And he's standing with our lovely plant care that we took with us so that our specimens didn't mold in that wet, rainy environment. So I just wanted to go over this fun story of the rediscovery of Licianthis Ceciliae. It didn't know that it was lost. I lost it. It was described from Veracruz by George Bitter in his 1919 monograph. It wasn't included in the Flora of Veracruz. when that was published in the 1980s. And I was having a hard time figuring out what it was because all I had was Bitter's original description. His type material had been lost in the bombing of Berlin in 1943. The type specimen was collected by the German anthropologist Eduard Seller and his wife Cecilia from Cerro de Chacomán volcano in the state of Veracruz in 1907. And it was given to Bitter to identify. And the species description just sounded so much like Licianthis prolifera that I just didn't know if it was something different, and I didn't have any material to look at. And I was looking at a lot of material of what I thought was Licianthis prolifera in Veracruz, and I identified as prolifera. And when I looked back at the species description of CE, I saw that he said that the corollas were stellate, not entire. And I looked back at my photographs of the specimens I had looked at from Veracruz at different herbaria, and I saw that the corollas were stellate, not entire. So I realized I had made an error. Thank goodness my publication hadn't gone to press. And I researched where Seller's duplicates from 1907 had been distributed, and I saw that a lot had been sent to Harvard. And so I wrote to them and I asked them if they had any Licianthis Ceciliae, and oh my goodness, I couldn't believe they had a duplicate in their regular collection. It was a duplicate of the type, and it had been sitting in their collection waiting for someone to look at it for 100 years. And so that confirmed my error of thinking Licianthis prolifera was in Veracruz, because it is Licianthis Ceciliae. And I contacted the botanist who had been collecting material of Licianthis Ceciliae in Veracruz, Gonzalo Castillo Campos of Inacol and Jalapa. And he graciously took me to his collecting site on the slopes of the Cofre de Perote volcano. And we found it in flower and fruit. And so that was a lovely story. So now I'm going to take us into Clade B on this whirlwind tour, and we're going to look at species that were placed by George Bitter in section Synanthoroides. And you can see right away that they're coming out in two groups, so they're not monophyletic. The ones in this top group are Licianthis Heterocleta, Geminiflora, Nitida, and Stephanocalix. And Stephanocalix was not placed in Synanthoroides by Bitter. It was actually placed in Ceres Prolifera, but I don't think it belongs there. And so in my paper on Ceres Prolifera, I talk about why it does not belong in that group. And in this phylogeny, it comes out actually with section Synanthoroides. And then the bottom grouping on this phylogeny includes Licianthis Synanthera and Sancticlari. So the best known of these species is Licianthis Heterocleta, which is a widespread species which goes from lowlands of Mexico down through, I believe, South America, certainly Central America. It starts out as a large herb with a green trunk, and then it can become woody at the base. It's shown here in a cornfield in Mexico, and the leaves are gathered as an edible green or calite. It has this truncate calyx with no appendages, which is typical of this section that Bitter together. Very few of them have well-developed appendages on the calyx. It's just a truncate calyx it's a little bowl. The coloration of the fruits is very typical of all of these species, with the maturation from green to red. And they all have stellate corollas and equal stamens. The coloration in Licianthis Heterocleta needs to be investigated because it starts out white up in Mexico and Guatemala and is mostly purple in Panama and Costa Rica. There's also variation in the amount of interpetallar tissue connecting the lobes. You can find it in some populations in Guatemala and Nicaragua. And so I actually think that somebody investigating this species for their PhD thesis would be fruitful, because there may be more than one species here. So a graduate student in Mexico, Miriam Reyes, looked at the morphology of these species that are placed in Synanthroides in subgenus Syngenensis. And she saw that the anther dehiscence is quite different in these two groups that came out separately on the molecular morphology. And so she also saw that morphologically. So Licianthis Synanthera and Sancticlari and Anomala all have fused anthers, and they dehisce by pores on the edges of the anther. And then the pores on adjacent anthers coalesce and make one huge pore. And they dehisce quite far down. So these are really gigantic pores. So Bitter actually described them as slits, but they really do remain these big giant pores. And he placed Licianthis anomala, this species right here, which has this type of anther dehiscence, in its own subgenus due to the type of anther dehiscence in calyx structure, which has little tiny appendages on it. Here's a specimen of Licianthis anomala. But these three are probably all very closely related. And then this other group, which comes out separately, which includes Heteroclita, Geminiflora, and Nitida. These have equal but unfused anthers, and they dehisce by little tiny terminal pores. So these are quite different. So the subgenus Synanthroides section Synanthroides may end up including Synanthera, Sancticlari, and Anomala, which all have the interesting anther dehiscence with the theca becoming confluent. And other species such as Heteroclita, Geminiflora will have to, which are in a different clade, would need to be called something else if someone wants to continue with those kinds of divisions. And here's just one last look at those two groups. So another group here in clade B that I spent a lot of time on is Ceres tricolor, which on this phylogeny is represented by Licianthis tricolor, Suritatensis, and Holocensis. So we published a paper on this group in Systematic Botany, and the species that I have included in the series are Arizolensis, Grandiflora, Holocensis, Michaelnii, Mario Valesii, Suritatensis, Tricolor, and Venturana. And the ones with asterisks I described as new. Bitter just called it tricolor because you can see that this is Licianthis tricolor, has a white corolla with purple markings and then these green dots that are at the base of the stamens. And these green dots are seen in other Licianthes species that aren't in Ceres tricolor. So it must be some kind of pollinator signaling, or it may have some kind of nectary. They're very shiny. I haven't investigated it, unfortunately, but it could use more investigation. But that pattern is seen outside of this series. And not all of the members of this series have these markings. For example, Holocensis is pure white, and Mario Valesii has very faded markings near the center of the corolla. But all of these species have simple trichomes, entire corollas, they're scandent shrubs, red berries, 10 appendages, and compressed seeds. In order to find Licianthis Mario Valesii, I traveled to Guatemala in 2017 and went out in the field with Mario Vélez and Luis Velázquez. And Luis found this location along the River Santa Ana near Asumidero, which is where the water from a river goes underground. As you can see right here, it was a really beautiful place. And we had gone there into the Department of Huehuetenango to also look for an elusive old species. And that is Licianthis cuchumatanesis, and it grows in these cloud forest areas. And it's based on 2 collections at the Field Museum made by Julian Steiermark in the 1940s. But unfortunately, the bridge to the location was washed out, and the local people were getting very suspicious of our truck. And so we kind of hightailed it out of there, and we did not find Licianthis cuchumatanesis. But we kept finding Licianthis Heterocleta, which is that species that I showed you that has interesting appendages that are obtuse and rounded and enlarged at the tip. So here it is with its stellate corolla and its fruit. And this species has these geminate trichomes with the whorls of rays on subsequent nodes that are very, very fancy. And they kind of look like little trees sticking up all over the stems and the leaves. And that's a wide-ranging species that's quite close to Licianthis cuchumatanesis. So they both share that trichome type. And during my visit to Harvard, where I was looking for Licianthis Cecilia, I really hit the jackpot and found more material of Licianthis cuchumatanesis. And so I was able to put together a decent species description for that that's posted on Solanaceae source. And this just shows how those crazy hairs look sticking up all over the leaves and the stems of Licianthis cuchumatanesis. And this is a drawing of the hairs that I made. So on this trip, I also was able to look for another species that I've called Licianthis freticaudia from Baja Verapaz, and it must be closely related. It looks closest to Licianthis chiapensis, and this is the hair type that that species has. This is the cuchumatanesis hair type, Ceteroxyloides and cuchumatanesis. This is the multangulate hair, very simple, of freticaudiae. And then I also have on this slide another species I described from Chiapas that I named after Dennis Breedlove, Licianthis breedlovii. He tried to get me to look at this like 25 years ago, and I was too busy with other stuff. And it's closely related to Licianthis hortulana. And this is the species that has really wild hairs that start out as multangulate stellate and then have further branching in the rays. And both Licianthis breedlovii and freticaudia had both been called Licianthis cuchumatanesis. So there's a lot of specimens out there at different institutions that say Licianthis cuchumatanesis that are not. And if you're wondering about the name freticaudiae, it's because Freddie Archilla took me out to find this species in Guatemala, and we made his wife, a physician, Claudia, late for work. So the species is named for both of them. So then I had to attack this group of species that I was quite scared of that has these geminate stellate hairs. And at the beginning of this project, I just couldn't tell them apart. They all look the same. But after a lot of hard work, I was able to separate them. And a lot of these species are not included in my monograph of Mexico and Guatemala because they are mainly down further in Central America. And they include such things as Licianthis Multiflora, which I really struggled with. And I decided to synonymize a lot of names into Licianthis Multiflora. Bill Darcy didn't include multiflora in his work of the Licianths of Panama and described a bunch of new species instead, and I synonymized a lot of those. And a lot of the names in Costa Rica, such as Ferrugina that had in Costa Rica, I decided to synonymize with multiflora. So I do have a list of the synonyms in my monograph of the species of Mexico and Guatemala. but I didn't treat that species fully because it doesn't actually occur there. It's in at the end of the monograph in species that were excluded for one reason or another. I also had to struggle with the difference between Licianthis Passiflora and Acetochondra in Panama, because again, I think Bill's view of those species differed from what I decided. I really struggled with it and decided that a lot of what he was calling Passiflora was actually Acetochondra, and that the morphology of that species needed to be broadened a bit. And then we also struggled with the widespread species Licianthis lenta, which is present from Mexico into Central America, Northern South America, and the Antilles. And Michael Nee realized that there was an older name for this species. And so, unfortunately for everyone, we changed the name Licianthis lenta to scandens. And we struggled with the morphological variation, especially in Mexico, in this group, and came up with two varieties, var scandens and var flavicans. And scandens is mainly Caribbean and is the real common form. It gets over into the Pacific side of Mexico a little bit. And then flavicans is mostly on the Pacific side, and the two intermix a bit in the lowlands of Nicaragua. And then the final project we worked on was in the latter part of 2020. We described 3 more new species from Panama that had just been sitting around in herbaria waiting for someone to look at them. And you guys, there's lots more new species of Licianthes waiting around for someone to look at them that just needs some tender loving care. And I hand it off to all of you to continue this work. So we described all of these species from Panama. And I'm not going to go into the old world species. Lynn could maybe answer some questions about that. Sandy Knapp is working on them. And I just finished with a picture of my late mother, Mabel, and her poodle helping me with my plants in the Lathe House at UC Berkeley during my graduate work. And there's my husband looking very happy holding up one of the species as well. So at this point, I'm going to stop the share, and it's 9 o'clock. So I'm going to have to hop off probably in about 5 minutes for my work meeting, but Lynn and I are happy to answer a few questions. I'm sorry that was so fast, and I'm sorry that...
No, that was great. Thank you, Ellen. That was amazing, the talk and how you show all the diversity of all the Licianthes. It's a great group. And I'm going to start with the questions. There are several in the chat. The first one is from Milo Pine. He says that he's interested to know if the sections of bitter nest clearly into the current clades.
So it's variable. I think that Lynn and I were just talking about that this week. So some of his, like Ceres Prolifera, it had, it was a fairly clear series, and I assume it will come out clearly, but it's not in the phylogeny yet. Tricolor, one of the species he placed in some other section, and he only had 2 of the species in the series. And I enlarged it a lot, so I think that, and then you saw section Synanthoroides is not monophyletic. So it's just a mixed bag. Some of them are going to be, I think, good groups, and some were just kind of a mess, like they were big grab bags. So.
Great, thank you. And then there is a question from Lynn, actually, if Licianthis Heterocleta have edible fruits?
Have what kind of fruits?
Edible fruits.
I think they're probably edible. I don't think they'd make you sick. A lot of Licianthes just kind of have insipid edible fruits, but I don't believe that's the emphasis. I think people are mainly using it as an edible green. In fact, one of Lynn's specimens from Chiapas, she actually wrote down on the label that it was used as a colite.
Okay, that's great. Then Franco Chiarini He's asking, are there any morphological synapomorphies to characterize clades A and B?
I don't think so. I don't think we've looked at that, but I don't think so. Clade A is a real grab bag because it has some of those Strigulosae, and then it has perennins, the Ceres Mesenodonte, and then it has those strange things from South America. I think those are, a lot of those are simple hair, but not all. Like, you know, they have, I don't think so.
Ellen, I agree with that. I don't think so either, or at least I don't know of anything that's really good. I've been thinking about seeds though, because some of the Licianthus clades have little tiny angled seeds. And so that might be a clade A, clade B distinction, but I'm not sure.
Except that those Ceres Strigulosae ones that are in clade A have flat triangular seeds, you know what I mean? They're not the three-dimensional chunky ones that are in Ceres Mesenodonte and the Rantinettii group.
Yeah, there's nothing that jumps out for me either.
There's so much work, you guys. There's just so much work for all of you.
That's amazing. Yeah, I can tell that like the seeds are quite informative, and there's a big variation in seeds inside Licianthes, which is amazing. And yeah, that for placement of the fossils is really informative, for example. So that's great. And actually also related to that question from Franco, I was interested to know, probably this is more to Lynn, if the chloroplast markers and the nuclear markers in the phylogeny are telling the same story or different stories about clade A and B, mostly.
That's a good question, Rocio. So I gave a talk on that had some Licianthese data in it, I don't know, last year. And so, you know, one of our big problems was a backbone and that one clade that's trying to hang around with capsicum instead of having a monophyletic Licianthes. But when we separated out, we got some genomic data, some transcriptomes from, I think, about 10 species of Licianthe capsicum. And it turned out the plastid phylogeny gave us a monophyletic Licianthes and a monophyletic capsicum. So that node, that problematical node, looks like it's variable depending on what genes and regions you are looking at. And it's probably incomplete lineage sorting that's leading to that little problem there. So, you know, even though the trees are showing the arrangement, when we combine these four genes, I don't think there's any way that I want to feel responsible for either getting rid of capsicum or combining part of Licianthes with capsicum, because morphologically speaking, capsicum and Licianthes are very easy to distinguish. So I know it's not following the phylogeny that I'm showing everyone strictly, but I think there's some really good reasons why we have trouble with that node. It's not showing up in every single tree that we analyze.
Okay, thank you. Yeah, that's interesting. And I think it's happening the same in different clades of Solanaceae as well.
Yes.
So,
Solanum has some issues too.
Okay, and of course there is a lot of congratulations from Judy Stone, from Janet Sullivan. Janet is saying that the hairs in breed lobby looks like the dendroid stelliform hairs described by Almut Seed in Physalis. This is really interesting. Also, Isabella is saying each flower and fruit more beautiful than the other, and what a wonderful history of Licianthes Cecilia. Thank you very much for the talk. Mario Vallejo Marin also saying congratulations and thank you. Also from Milo, from Gloria. Stacy says that she would like to know about the nectar. She thought that they were all vast pollinators and didn't make nectar, but those shiny spots. It's kind of-
No, they look like extrafloral nectaries, but I really haven't investigated them. And it's not hard to grow these shrubs in the greenhouse. I mean, it would take a few years for them to mature, but I think it's really interesting to, I mean, they're either just pollinator signaling or they're actually exuding something. I've never seen droplets. So
that's interesting as well. There's so much to do there. Yeah, it's amazing. Clara Inés Orozco, she's asking about if there is a relationship between the habit of the Licianthes and branch or simple trichomes, the presence of that different type of trichomes.
no, because in the perennins, the Ceres mesenodontae, those herbs, you can have branch trichomes or and simple trichomes in the same population. Like you can have individuals with just simple trichomes and individuals with branch trichomes in the same population. And you have species in that group that range everywhere from multiangulate stellate to branch to simple. You have shrubs and vines like in Ceres tricolor that have simple trichomes, and then you have shrubs and vines that have very complicated types of trichomes. So the epiphytic plants that are in parts of section Synanthoroides, those also have everything from species from just simple trichomes to species with branched or multiangulate stellate trichomes. So it doesn't follow the habit.
Okay, and Clara Inés also has another question. If it's easy to find differences between capsicum and Licianthes in vegetative state.
Oh, if there's no flowers or fruits?
Exactly, yeah.
Actually, it can get hard if there's no flowers, because if all you have is fruits and you have a calyx with appendages, you have to then really know your species, because there are some Licianthes with 5 appendages, and the capsicum usually have 5. So if there's no flowers and fruits, I would have a really hard time. How about you, Lynn? You're muted.
Yeah, I'd have a hard time, but you have to remember that Licianthes is way, 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 way bigger in terms of species than capsicum. So for me, it's simpler to get a handle on which capsicums you might expect in different spots. And, because there's maybe 40 species in total. But Licianthes, there's lots and lots. But I will say that I think that a lot of vegetative capsicums and Licianthes look alike.
Well, thank you. That was the last question. Also, there are more comments about nice presentation, and also Gloria saying that yes, it's very difficult to distinguish capsicum and Licianthes in vegetative state. So if someone else has a question, like you can type, or yeah, there is another question here from Isabella. She says, does the difference in trichomes reflect the phylogenetics in Licianthes?
I don't think so. I mean, I think that kind in Breedlovii, that's so complicated. I only saw that in Hortulana and Breedlovii. So it was confined to those couple of species, but in terms of broad phylogenetics, I don't think so. What do you think, Lynn?
Well, I don't think so either. I think that some of the clades have distinctive hair types, but they're all mixed up on the cladogram. It's not a really strict relationship of, a whole big clade has stellate hairs or not. And then you mentioned that branch and simple hairs can occur within a single species.
Within a population, yeah.
Yeah.
Just to make it hard. Yeah,
that happens a lot with hers. And what morphological character would you say that is the most informative to distinguish different clades within Licianthes?
I don't think it's like a single character. I think that within each group that I looked at, it was a combination of characters. So it would have to do with a combination of the corolla type, the trichome type, the habit, the ecology, and the fruit and the seeds. Like it was sort of a combination of things, and altogether it supported a group that looked like they were cohesive.
That's right.
Also, I think it's important whether the stamens are equal or not.
That's interesting. It's kind of in different groups, like far related within Solanaceae. Always the androecium, the stamens are quite informative. So, but there is a lot of conversions as well, for example, in Eucramina. So, yeah, that's kind of.
Yeah, I agree with Lynn. Stamens are a nice thing to look at first. Trichomes and stamens.
That's great. Okay, I don't know if we have any other question. Remember that you can type question and I'll give you the words. So if you want to comment or ask a question directly, just type. Okay, here is Shay Goldberg. What is the benefit of the crazy hairs? Are they defensive?
No, because that particular species with those wild hairs, I mean, they grow with other Licianthes in the same habitat that have simple trichomes. Does everything have to have a reason? I'm not totally sure that things in life aren't sometimes random and then they get stuck in place. I don't know what that was about. They're just. And then the crazy hairs and Ceteroxyloides and Cucumatinensis, and in that cloud forest area, you have other beautiful species that have simple hairs. So I don't know what the advantage of that would be. I certainly would think that those hairs would help gather moisture from the clouds, but I don't know how important that is, or what, if it's an insect relationship or what. I'm going to have to hop off, you guys, but I did give my email on the first slide. And if anybody needs anything, please let me know. And I hope other people continue studying this wonderful genus.
Ellen, before you go, I have a comment for everyone. I just wanted to say I've known Ellen for many, many years now and say how much I've enjoyed knowing her and working with her. And I encourage you to look at this beautiful, beautiful publication on the Licianthes of Mexico and Guatemala. It can be an inspiration for any of you who are doing taxonomic or floristic treatments. It's just gorgeously done. And I want to reiterate what Ellen said several times in her talk, but there's so much more to find out about Licianthes. There's lots of species, new species out there, a lot of things that we don't know about it. And we're getting to be pretty old ladies now. So we're handing off the torch gradually to all of you youngsters. And if you want some good problems and interesting things to work on, there's a lot of things to do with Licianthes.
Yep.
Thanks, Ellen. That was great.
That's great. It was a great talk. And thank you, Ellen. It was amazing to have your talk here. And yeah, so I'll email you if there is additional questions. I email you the chat and everything, so you can check them later. Bye. Okay, yes, there are more comments from Isabella. I don't know if there is any other question. I know that we are running out of the time, but yeah, thank you. Very nice and useful talk from Clara Inés. So I think there is no any more questions. We are finishing the seminar. Next week, next Friday, we are going to have Susan Strickler in the next talk. So stay tuned. We're going to send the email on the information very soon. So thank you, everyone, and see you next week. Thank you, Lynn.
Thanks, Rocio. Bye-bye.
Bye.